So this is a study of Alaskan paleosols and permafrost. And I'm part of the Army Cold Regions Research and Engineering Laboratory. And my collaborators are from the Environmental Laboratory and UC Santa Cruz. So this is a beautiful picture of Denali that is just reminiscent of beautiful Pikes Peak out here. So just a little bit about the research portfolio at the Cold Regions Lab. It's not limited to Cold Regions research that we do. We model soil processes for threats on surface soils. We're interested in dust, so we actually have field campaigns in the desert Southwest, which is very different from Bare Alaska. We look at bioremediation in cold environments, and what we'll talk about today is DNA tracking. So soil is quite diverse. 99% of the organisms actually cannot be cultured. This is the site scale of the forest in Fairbanks, Alaska, which is our study site. And as you tromp through the woods, you fall down a lot because they're tussock tundra. But also, you don't know where permafrost is. There's discontinuous permafrost. So if I had my auger and I was tromping around and decided to sample, I don't know if I would hit permafrost. I don't know the age of that permafrost. But if you dig a soil pit and you see the permafrost core, there are different layers. And that's because in Alaska, loess blew in and created a stratigraphy in the environment. So we use molecular approaches to study bacteria, archaea, and fungi. And we hypothesize that there's differences in microbial composition because the depth of the permafrost represents a certain age. So the complexity of this, particularly in soil, is that microbes are interacting with one another. And this is on spatial scales, but also on temporal scales. And that just blows my mind. And it's really, really hard to track. So we also visualize this complexity, and it's probably difficult to see because it's so wonderfully bright in here, but between the soil particles, there's tendrils of fungal hyphae that are connecting them. So we use DNA, but it's also helpful to actually visualize what's going on in the soil pore, and that's a scanning electron micrograph image. So I'm going to take you back in time through the Holocene and then the Pleistocene, which is about, the late Pleistocene is 60,000 years before the present. So to me, that's ancient. To my paleoecologist friends who study the Jurassic period, it's not. But I'm just a soil microbiologist, so I'll use that. So about 55,000 years ago, this is a neat cartoon of megafauna, bison, horses that were roaming the landscape. There's dry tundra and forbs, which are herbaceous plants. At about 25,000 years ago, there was the last glacial maximum, which means that glaciers were at their greatest extent. And interestingly enough, you can see Greenland, which is still fully, almost, well, it's melting, but there's a lot of glacier there, obviously. But interestingly enough, in the top, that's Alaska. And in interior Alaska, it was actually never glaciated, which is great for us because the loess blew in and we don't have that manipulation. So the idea is that we can track what was there at the time the feature formed. And that was a cool, dry climate until the glacier started melting. And that resulted in some moist tundra, and that occurred about 17,000 years ago. Then the megafauna died. So there's the fossil example there. About 3/4 of the animals died, and it's really not sure. And I have to say, when going in the literature, I didn't know what happened in the past. I never thought I was a history buff, but I'm becoming one. And so there's a lot of controversy of what really happened. And think about it, what do we really have? We have bones, we have the chemistry, and we have fossilized pollen to try to track it, but we also have DNA. And then going into the Holocene, which is the past about 12,000 years, you can see that in the Pleistocene there was these big broad scale temperature swings, and then the Holocene was somewhat stable, up to the Anthropocene, which is us impacting. And the vegetation in that region in interior Alaska changed quite a bit, from herbaceous tundra to an alder-dominated forest to tundra and boreal forest. And you could think, who cares? Well, with these broad changes, that actually attracted different animals to the region, and as we all know, microflora are intimately tied to vegetation and then drive the larger animals. So our objective was to study the relationship between these biogeochemical pieces of evidence and relate them according to these really big climate change events. And why does the Army care? Well, it's using DNA as a tracking tool, number one. And number two, as our climate is changing, it's what we have to forecast what might happen. In the past, different plants interacted together that you wouldn't necessarily see, and then we can try to predict that. It's kind of all we have right now. So I'm first going to take you through our Holocene, which is our modern surface soils, and our wonderful garb is the white bunny suit that we affectionately wear because we're also tracking human signatures. And then we have a bluff, which is on Fort Greely in Alaska, which represents a really broad timescale of the Holocene. We were lucky to stumble across that. So the first conclusion is that modern soils don't represent paleosols, which is what we expected, but it's nice to see it. I have another study in Barrow where we're growing plants in one place, not in the other, and the microbial communities don't differ at all. So this was really exciting. So on the left, for the bacteria, those are the modern soils. They're different from the paleosols. And we also saw the same community structure in the fungi, but to a lesser extent. Now, I also, being a soil microbiologist, am very interested in how abiotic variables drive these systems. So we took a deeper look just at the paleosols. And interestingly enough, what the samples collected earlier were different from what was later. But on top of that, drivers like pH, organic matter, water content that would typically drive the system weren't. So there wasn't a correlation under canonical correspondence analysis. We also looked at different organisms. So I'll take you through this on the left. This is our modern surface soils, and then we go from about 300 years to about 7,000 years before the present. So you can see different tephra, which are the volcanic eruptions and the ash that deposited, and then the different soil layers that represent the different time periods. So here are about 8 different bacterial phyla that represented statistically significant differences across the time frame. And so, for example, proteobacteria are more abundant in the modern soils as compared to the older soils. Firmicutes, which are our spore formers, were more abundant at Paleosol 3, which is about 3,000 years before the present. So you can see that there are broad community shifts, and something interesting is that at Paleosol 6 and 7, there was a thermal maximum of warming. So the climate got warmer, and you can see some broad shifts, like the abundance of the Geminodides, which are soil-borne organisms. They're oligotrophic. So just as the built environment doesn't have much going on, one could argue that's the same for soil. They're just starving there until maybe a beetle dies next to them, and then they all become active. So only about 10% are active. We also looked at the fungi. The first 5 of them are from Ascomycetes, and then the last 3 are Basidiomycetes. So Basidiomycetes are typical mushrooms, and what you can see here is that in Paleosol 2, which was a little bit more recent, there was a thermal cooling, and that things like Agromycetes, which are decomposers, have a versatile substrate utilization. They are actually more abundant, so on the Basidiomycetes side. So it gives you a sense of, hey, this is Paul. Paul likes to eat meatballs, but Robyn likes to eat Brussels sprouts. You can start inferring the activity without actually doing the metagenomics, depending on what your funding situation is. So we did take a metagenomic approach, and this is a neat cartoon of the Holocene. And we saw an increase in genes relating to fatty acids, virulent, which is something that as the landscape changes, different microbes might emerge and could or could not become virulent and pathogenic. Carbohydrate metabolism, which all that label carbon and the microbes become active, that will certainly impact global change and larger organisms. Aromatic degradation is quite curious because in these areas, especially where soil is locked up, but contaminants progress, these organisms have the genetic versatility to actually break down these aromatic compounds. So let's go back even farther. And this is still near Fairbanks, Alaska, and it's the Erdic Permafrost Tunnel, which is a really incredible resource. The only other one I know of is Russia, and for the Sochi Olympics, they opened up their permafrost tunnel. I said, no, you're contaminating it with all the human, the pig pen cloud? So I cringe, and kudos to us because we take tours, but we don't try not to contaminate it. So this is us, again, in the bunny suits trying to collect samples. And this reaches about 60,000 years before the present. So the neat thing is the permafrost tunnel was created to understand geophysics. In the Cold War, and we're posturing towards Russia, we started building roads and thought, why is it undulating? It's because there's something different underneath. It's called permafrost. And so this tunnel is typically utilized for geophysics research, and this is one of our first opportunities to study the biodiversity. And when you go there, it's really funny because they're so laissez-faire. Here's a mammoth bone. It might be 30,000 years old. I can touch it with my own hands, and you walk through, and the permafrost is sublimating, and you can see bison horn. So there's evidence of these fossils that these organisms existed. And so when you walk into the tunnel, it's almost like you're walking in time. And again, my geophysicist friends will cringe because it's not, I'm saying lust blew in, but it wasn't in this very consistent way. But over the course of tens of thousands of years, you can have discrete differences in age. So I feel shameful that I only have like 10 points on my PCOA plot. Thanks, Andy. But anyway, there were a hard 10 points to collect. And what we can see up here, so this metagenomic analysis, and you can see the bacterial dominance. So you're saying about the built environment, it is so important. It's important for anything. Bacteria are everywhere, and they are the best chemists. So here are just 3 different points in the tunnel representing different ages from about 20,000 to 35, 40,000 years ago. And you just see a lot of bacteria. The other important note is that you have to squint, but our extraction blank is far away from our samples. Now, I thought working with soil was difficult. Permafrost is like, how shall I say, eating the Tootsie Center of a Tootsie Pop without licking it. And so it really took a lot of energy to try to figure out creative ways to get into it. And so I wouldn't be saying up here, and we discovered alligator 40,000 years ago. I don't think so, not in Alaska. So that's a really, really good point. And so we see separation in the bacterial communities according to this age, and we have the metagenomic analysis that we're going to use to further understand larger functions. So here are again the bacteria, and what I want to show you here, this is a cartoon representation of the tunnel, is that there is a core paleo microbiome that's present, and proteobacteria, actinobacteria, these are the common soil players. But diving a little bit deeper into the class level and what these organisms are capable of, and I really like what Jack had mentioned about looking at the rarer community members and seeing how they might emerge as the permafrost thaws, because not all permafrost is created equal, as we're learning. So again, our cartoon of these megafauna roaming the earth, and so we see an increase in plant alkaloids, so these materials created by plants that bacteria will utilize. Tetrapyrals are utilized by cyanobacteria. Interestingly enough, cold shock proteins are low, and I should have mentioned a little bit earlier is that one criticism of permafrost research is that these bacteria, these microbes might be active. Well, we did a little respiration experiment, and it would take about 492 days for them to respire 1 gram of carbon at that temperature. So they are active, but they're pretty pretty slow as compared to our modern soils, which are about a day or less. So microbes are dominant sources of DNA, so we're developing and working with others to create unique strategies to look at other larger organisms. The ancient climate shaped these fauna and flora, and we're looking deeper into trying to resolve the different interactions. Actinobacteria have these bioactive metabolites. So the question could be, could permafrost be the new Amazon, where new materials, new organisms are created? I don't know if you've heard about the story in Siberia where permafrost thawed, the caribou infected with anthrax, yeah, Captain Dancy shaking his head, and then people were they died. So we want to avoid that. And the proportion of what was there then is not what's there now, but we might be progressing towards that in the very near term. So I just want to thank the Assistant Secretary of the Army for funding our research. Who knew that the Army could fund permafrost microbiology? And the Fort Wainwright archaeology program was pivotal in this research. So, thank you.